Let's resume the afternoon session. The next speaker is John Mackenzie from the University of Strathclyde, and the talk is about computational models for single-cell migration and chemotaxis.
Okay, thank you very much. Good afternoon, everybody. First of all, I'd like to thank the organisers of this very interesting workshop to allow me to present my work. As you can probably tell from the opening slide, I'm a mathematician. As Charlie said, that means that we have the people to thank on the first slide rather than the last slide. And the other thing is we're going to spare you the mug shots, so you never. Oops. Oops. What am I doing?
I think you turned off the projector.
No, you shouldn't do that. You should wake up the screen.
Ah, there we go. Yeah, okay. One bit on. Okay, so the people I want to thank are a couple of PhD students of mine, Michael Nolan and Grant MacDonald, who are in the Department of Mathematics at the University of Strathclyde in Glasgow in Scotland. And my background is really in numerical analysis, so the development of numerical methods for solving PDEs. And I had no background in biology until about 5 or 6 years ago, when Rob Insall, who's a cancer cell biologist at the Beatson Institute for Cancer Research in Glasgow, came to the department looking for a little bit of help to do some modelling for cell migration and chemotaxis, and immediately was looking at some of the initial movies of what was going on. I really got interested in this particular field. So how do cells move? The movie's not been seen before, but I'm fairly sure everyone here has probably seen this movie somewhere. And in fact, something very similar to this has appeared at least on 2 other talks so far. So here we've just got an example of a neutrophil cell. It's migrating towards this little bacteria, and you can see that it's migrating by forming a pseudopod or lamellipod here at the front, and dragging along the rest of the cell behind it, and it's also shoving these red blood cells out of the way. And the thought on how cells actually move is this sort of continuous cycle of the cell forming actin-based protrusions at the front of the cell, attaching to the substrate. The cortex is then under tension, and then there's a deadhesion at the trailing edge, possibly mediated by myosin cross-linking with actin. And this leads to directed cell motion. Okay, so chemotaxis again probably doesn't really need to say too much about that. It's obviously just the directed cell motion in the presence of some cue, some chemical cue. So here we have some dictyostelium cells migrating upwards here. And again, the interesting thing is that at the front of the cells, you can see these little pseudopods, these protrusions shoving their way out, and at the back of the cell, it's a little bit more rounded morphology of the cell. And this movie here is the sort of infuriating micropipette experiment, similar to what I think Rob had on this morning. Here, the F-actin isn't sort of labelled, but you can see the cells, these dictyostelium cells, again migrating quickly towards the source of the micropipette, which is emitting cyclic MP that cells like to migrate towards. And again, the other thing that's interesting to note is that the cells are doing this by generating pseudopods. But again, some of the cells, when the micropipette has changed direction, don't necessarily just immediately depolarize. They sometimes change direction by almost by doing a U-turn. And so these are some aspects that we would like to incorporate within our mathematical and computational models. So I got into this area after, as I said, Rob Insall came to our department, and he just recently published a paper looking at migration by pseudopod bifurcation. And this was essentially an experimental observation that a lot of cells, and in particular dictyostelium cells, migrate by generating pseudopods at the front of the cell, but those pseudopods often bifurcate and split into two. And this bifurcation essentially allows the cell, I guess, to explore different areas. And in terms of chemotaxis, it's often just the most upgradient pseudopods which tends to survive and win out. And so this interesting behavior had been observed in a number of different cell types. And the idea was basically, could we construct a mathematical and computational model that maybe had some of these, this sort of behavior. So generally what was observed was lateral pseudopods, which essentially are pseudopods getting shoved out at the side of the cell, where rarely observed that pseudopods tend to be generated where previously generated pseudopods were. And again, that sort of seems relatively obvious because there has to be a movement of the actin machinery to generate pseudopods. New pseudopods tend to be generated by bifurcation of existing ones, and they tended to be generated at regular intervals independent of the orientation of the cell. So, given those observations, and interestingly, in the timescale of this pilot project we had 6 months to get a postdoc and try to get something up and running. And so rather than basically going down to scratch, we basically thought, okay, so what can we do here? Well, let's model what's going on in the cell cortex, cell membrane by some set of what seems at the moment rather arbitrary reaction diffusion systems, but the idea was basically, can we get pattern formation type behavior that coincided a little bit with the experimental observations. So this is really, at the moment, a little bit of a top-down type model. It's an activator-inhibitor type model, but what's a little bit more unusual to the standard models that use, for example, in describing patterns on leopard skins for spots, etc., is that this system of equations is highly dynamic. Of course, if we're going to try to use this in pattern formation in cells, which are highly dynamic, of course, the last thing we really want is the system to fall into steady states. Okay, that's obviously a disaster for a cell, so this needs to be as dynamic as possible. Okay, so this system was put forward by Hans Meinhardt a while ago. I'm sure it was never intended to be used the way we ended up using it, and it's actually a 3-equation system. Okay, so A here is a local activator, and if you want to hang some sort of biological entity on that, you can maybe think of this as some actin nucleating factor, whichever one's your favorite, SCAR, whatever, leading into ARP2, 3, basically something that's going to lead to actin polymerization. It's not necessarily going to be actin. It's going to be something that helps nucleate or polymerize actin. So this basically is evolving on essentially in the cell cortex. It's a reaction diffusion system of equations. So what's a little bit unusual here is that these operators here, so this is essentially diffusion. So this is diffusion that's actually just taking place on the cortex. It's not taking place necessarily here through the cell. So this is essentially like the Laplace-Beltrami operator. It's like a surface operator. This is a surface divergence, which is necessary because the cell is actually moving. So we're assuming that the material velocity of particular points on the surface are moving with velocity u. And here we have some nonlinear kinetic term coupling the activator, the global inhibitor, and the local inhibitor C. Okay, so I'm not going through all the details of this particular model. Roughly, A is autocatalytic. So basically, A feeds back on itself. So this again seemed attractive in the sense that this was going to give us bifurcations of our pseudopods, and also new pseudopods were going to be formed where old pseudopods were. So this autocatalyst seemed to be built into this particular model. The role of B essentially is to make sure that pseudopods are not generated everywhere. And again, that sort of coincided with the experimental data. And in a way, the C here is a local inhibitor. Now, the role of this in this particular model is to ensure that where we get peaks, localized peaks in A, which is going to result in actin polymerization, we don't want those peaks to live indefinitely, because the cell has to reorient itself, it has to be dynamic. And so that's done by the inhibition of C. So C is basically formed in the presence of A, but at a slightly slower rate than A, and it basically inhibits A eventually. So what that tends to do is it tends to poison any peaks in the activator profile, thus allowing new pseudopods to be generated elsewhere in the cell, but ideally that's likely to be close to where the previous pseudopods were.
So do you have a preferential range where pseudopods are going to form, or it's an emergent process of the reaction diffusion system?
Okay, so the other term I should say in this particular model here is S. So this is going to be a signal term that's going to be related to a very simple model for receptor ligand binding. So this is where chemotaxis is going to come in. So in the absence of S, this model will still generate pseudopods because certainly for dictae, cells still migrate in the absence of any chemotractant, and they do so almost randomly, but they still exhibit a persistent random walk. So they still have this same behaviour that the pseudopods tend to be generated close to previously generated pseudopods, but the initial direction would be random. Okay, but chemotaxis is fed in through this term S. So S feeds into the production of A, and I'll talk about S in a second. Okay, so basically the general hope here was that pseudopods would be generated even in the absence of any chemotractant. So if S just turned out to be 1 here, we would still have a pattern formation system. We have restricted active areas of the cell. This is due to the global inhibitor. So this is somehow building in the idea of cell polarization. There's parting in time to allow the generation of new activated areas so it doesn't fall into a steady state. And the ability to reorientate in terms of a signal, it comes in through this term S. Okay. So what we built into the model was a very simple ligand receptor binding model. So we're essentially assuming that ligand molecules bind with receptors on the plasma membrane, forming some complex. They dissociate with rates K1, K minus 1. In our model, we're going to assume that the total number of receptors is going to be constant. And so basically we're going to be able to calculate a local receptor occupancy, which is essentially just the ratio of the local total receptor number and those that are occupied. So that's a system of reaction diffusion equations which are going to be solved, and I'm going to tell you how you do that numerically, on the evolving cell membrane / cortex. Okay? So what I'm going to do now is I'm going to couple that to a very simple model for the diffusion of ligand molecules external to the cell. Okay? Now, a lot of the machinery I'm going to talk about as far as the numerics is concerned could be used for modelling intracellular processes, but of course that's far more complicated. But if you have a favourite model for how to do that, then at least the numerical method I'm going to show you can in theory be applied, but I just want to show you it applied to this somewhat simpler situation. So I'm just going to assume that ligand molecules external to the cell are going to diffuse by standard linear diffusion. So that's L here. The concentration of occupied receptors is denoted by LS. So that's going to evolve on the cell membrane. So I'm going to assume that there's this binding and unbinding process that's taking place on the membrane, and this also allows potential lateral diffusion of receptors on the membrane. So there's a diffusion coefficient for that as well. So I have to have a flux balance equation at a boundary condition on the cell membrane as well. And so this is the diffusive flux of molecules onto the membrane. This is the binding and unbinding. And this term here, which looks a little bit odd, is an advective-like flux that's caused by the movement of the cell. If we assume external to the cell, the region, the material points there are stationary. So this is a cell moving through some stationary domain. And this is what's sometimes called something like the windshield effect. So if you can imagine you're in a car and you're driving through the rain, of course, the front of the windscreen experiences a higher flux of rain drops onto the windscreen in the back of your car, and that's affected by this term here. So the front of the cell, depending on the size of you here in relation to everything else, there's going to be a flux of molecules onto the moving boundary, and conversely a flux off at the back end of the cell.
It's a jump.
Yes, yeah, because there'll be a jump because you outside will be 0, but you on the boundary will be non-zero.
Okay.
Okay, so the chemotactic signal term that you saw in my original set of equations is just going to be based on the local receptor occupancy. So if I solve this coupled system of PDEs, I can work out the local value for LS here. The LS allows me to calculate local fractional occupancy. That gets fed into my Meinhardt system, which has all been solved on the evolving cell interface. So that's the model. So I'm going to move my cell. So how am I going to move it? So again, a lot of I'm going to have some assumptions here. So I'm going to assume that actin polymerization creates some protrusive pressure and some outward normal velocity of the membrane. So basically where A is large, I'm going to push my way out. Obviously if I do that, then the cell's just, I don't have any control over the cell area or the cell volume. And so I'm going to assume that the volume in 3D or the area, a lot of what you're going to see is in 2D, I'm going to assume it's going to be approximately constant. In my model here, I'm not constraining this as a hard constraint. I have a sort of loose way of controlling the area. So I essentially assume that the normal velocity at the membrane is given by this outward protrusive pressure due to the presence of A. And I'm also going to have this sort of cortical tension effect, which is going to keep the cell area constant. So here, where the curvature is high, the cell is going to pull itself in. And where the curvature is low, this effect is going to be relatively weak. This lambda factor here is just a control to ensure that the total area of the cell is approximately constant. But as I said, it's not a hard constraint here. The area can go up slightly and down slightly. Okay. Right, so that's the model, and I'll show you a little bit later the simulations of that model. What I want to talk a little bit about is how you would solve those sort of equations. So Charlie touched on this a little bit yesterday. So we'll be solving reaction diffusion equations on moving/growing domains. So here we have an interface between these in this region here. So I have omega of t. This is omega of at some later time. So here's my cell moving. I may have material points in the extracellular region moving with a particular velocity, or the points may be fixed. But this is the general setup that I've got. And what I want to do is I want to solve these differential equations on this boundary, and I also want to solve the differential equations exterior to the cell. And as I said, a lot of the machinery can be used to also solve differential equations in the interior of the cell. So my general type of equation I'm going to solve is going to be of this particular type. So again, this is just a scalar reaction diffusion equation taking place in the bulk. And if I have a bulk velocity u, then I'd have this additional term here. If there's no velocity in the bulk, then of course that term just disappears. If I have growth or movement, then potentially I have two additional terms from here if I expand this out. One which just gives me a sort of passive transport of C, and one, depending on the divergence of the velocity field, will either give me a local dilution or concentration of my chemical C. In my boundary domain, again, I have this sort of rather general form of my reaction diffusion equation. So these are my coupling terms, and this is any potential nonlinearities that I have that are purely due to taking place in the surface. And again, these operators are these surface operators, not your standard Cartesian divergence and Laplace operators. Okay, and here we have flux balance again, as stated. Okay, so one of the main issues of solving PDEs on evolving domains is that you really need grids. Okay, you need a grid to cover your particular domain. And although maybe grid sounds rather dull, it's pretty important in this particular context because you really want efficient, good methods to solve these problems. Okay? And grid generation tends to be pretty non-trivial. Also, you may think that because you have a velocity field at your disposal, that sort of tells you how you should actually move your grid points. However, that would give you a sort of Lagrangian way of moving your mesh. But often that's far from ideal if you're want to solve PDEs, because usually a mesh can become very distorted, very poor quality. And so the approach that we've used is this so-called ALE approach, this arbitrary Lagrangian, where there's an arbitrary velocity field that you construct, which basically comes through your grid generation procedure. And this velocity here is denoted by W. Now, when you're reformulating your PDEs, in terms of this moving reference frame, then you have to include the effect of this term W because, as I said, it's totally artificial. It's what comes in through the grid generation procedure. But if you don't take consideration of this. You will create artificial velocities when you're solving your PDEs. Okay, so you have to take care of this, and I think there were some questions on this yesterday. You have to be a little bit careful about how you do this, because if you do it with, not carefully, then you can introduce artificial source and sink terms in your equations, and of course that's the last thing you really want to do. Okay, you want everything to be sort of rather invariant to this ELE velocity. Okay, so the PDEs are going to be solved by a finite element method. Again, the finite element methods are good for complicated domains. And the other advantage in this class of problems is that the boundary condition between this flux boundary condition is incorporated very naturally within this formulation. And so this is a nice framework in which to work in. So it looks rather complicated, but the general idea is that we multiply through by suitable test functions and use integration by parts. The integration by parts is the thing that deals nicely with this flux boundary condition. And basically you have a set of equations for the bulk domain, set of equations for the boundary, and here we can see the ale term being incorporated in both of these equations. Okay, so here the W is the ELE velocity, which comes from mesh generation. The U is the physical velocity that would come through the velocity of the material points. So again, these terms, for example, would completely disappear if your grid generation scheme was Lagrangian. Okay, but normally you don't want to do that, so you have to take care of this. The other thing is this is a little bit unusual because this is a sort of conservative form of this scheme, and again this is important if at the discrete level you want to make sure you're conservative, so you conserve mass in your system. Okay, it's non-trivial when everything's changing as a function of time. Okay, so after discretization, what happens is you get a large system of ODEs for your unknowns essentially in the bulk, and which are coupled to a set of ODEs for your unknowns which are on the surface. And we've proposed a very simple sort of iterative scheme here for dealing with the solution of these processes. So basically we just predict the solution on the surface. So we're assuming we know where the surface is at the forward time level, so we can evaluate all of these particular matrices. So we predict the solution at the forward time level. We basically update the solution in the bulk using a so-called Crank-Nicholson scheme. This is just a scheme which is basically second order accuracy in time. And then the prediction stage initially is just a first order backward Euler scheme, which is stable but only first order in time. So to correct the solution on the surface to make that second order as well, we go back and we correct the solution with a Crank-Nicholson scheme. So it's a sort of predictor-corrector method where we predict on the surface, we solve in the bulk, and then we correct on the surface. So the whole idea here is to ensure that the overall scheme is second order accurate in time and stable. And if we're using linear elements in space, we expect that to be second order in space. Okay, so here's a little model problem. So here, true to my numerical analysis roots, we pick a problem that we know the solution to. Essentially, it's just solving the diffusion equation in a unit circle. We have an equation on the boundary, which again is a sort of like a heat equation here, and there's this sort of coupling term on the boundary. So basically, there's molecules of the bulk that can go onto the surface and diffuse. And it turns out for this particular problem, there are infinitely many solutions, but the one in which we actually choose is this particular one here, which can be written in terms of Bessel functions. So this isn't phenomenally interesting from a cell biology point of view, but the idea here is at least to make sure that the method that we're going to use for a much more complicated situation is being validated at least on problems that we know the solutions to, and also just to check the rate of convergence of the method. So looks as if it's doing a reasonable job on the surface, but numerical analysts are always happy with seeing these type of plots here. So if we let the number of elements become larger so that the mesh size goes to 0, what we're looking for here is the slope of this to be 2, which tells us that both the bulk solution and the surface solution are converging at the rate we would expect with linear elements. And similarly, if we do a convergence study in terms of the time steps, so we take more and more time steps, that we do get this second order accuracy that we were hoping for. And if we didn't do that correction step in the Crank-Nicholson method, this method would only be first order in time. So that correction step takes us back to second order accuracy. Okay, right, so the method, at least for that model problem, looks reliable. So now what I want to talk about is how do we generate the meshes in time. Okay, so the idea here in a lot of mesh generation is you can think of a mesh on an evolving domain as being some mapping of a mesh that's covered some fixed domain. And there are a number of different ways in which you can go about doing this. The one that we're using is based on variational formulation here. So basically we're assuming that this mapping somehow minimizes this functional, where G here is some, in 2 dimensions, 2 by 2 symmetric positive definite matrix. The interesting thing is we can, through the choice of G, we can actually get some degree of adaptivity of our moving grid as well. So the grid's not only going to move and cover the domain, you may also have the facility to adapt the grid to solution features. So for example, you may have large gradients in proteins close to the membrane. And so to pick those gradients up and numerically, you may need to do some degree of mesh adaptation. So simultaneously, your domain is changing shape, evolving in time, and you also want to do adaptivity.
Question?
Yep.
So this is a global formulation.
Yep.
But frequently it's very locally that your grid has to be changed.
Yep.
How do you balance this idea of a local mesh quality versus this global functional?
Yeah, so it's all through this matrix G here. So the mesh is thought of, if you want to do adaptivity, you can think of equidistributing some weighted adaptivity criteria. So it's globally equidistributing, but because that function is local, that gives you the local mesh spacing.
So this G is actually space dependent.
Yeah. Okay, so here you go. So some examples of G could be something that's a diagonal matrix. So for example, you could have large gradients in G. You want this to be positive, so something that's based on the gradient could work, something possibly based on the Hessian of C, so something dealing with second derivatives. And if your problem is really complicated, you might just be quite satisfied with adapting to features somehow in your particular system. Now, we need to create for robustness, it makes sense to, rather than minimize this function directly, we've got a time-dependent problem. Instead of taking steepest descent direction, we evolve the mapping in this particular way. So we introduce a temporal smoothing parameter and some other parameters just to smooth everything off. But the long and the short of it is we have a system of PDEs that we also need to solve for the mesh. Okay, so it's not necessarily that you get this for free. There's some additional work that needs to be done. Okay, so we get this coupled set of parabolic PDEs to solve for the mesh mapping. So here's an example of the generated mesh for something that looks like a simulated Dicty cell. So you can see that the boundary of this domain here is coming from a numerical simulation of that system of PDEs that I showed you earlier, and the interior mesh has been generated by the system that I just showed you. So here there's no adaptivity. This is just a mesh which has some sort of smoothness properties in the interior of the domain. But you can see clearly that the mesh is certainly covering the domain, and the domain is evolving as a function of time. Now, we can also do simulations with this method so that we can adapt to particular features. So here, for example, we may have,
A quick question in terms of the pseudopodia.
Yep.
Am I seeing it rather as like a little oscillation before it chooses
Yeah.
one direction?
Yeah, absolutely.
Where's the oscillation coming?
Well, it's coming. Well, I'll show you a little bit later a simulation where you'll actually see the activator profile, and you'll see it splitting, and that's where you get this sort of pseudopod splitting. Okay, so here we have the grids generated. So here the grid is actually adapted to the membrane. So this sort of gives you this almost like 3D effect of what's going on. Here we have it adapted to some arbitrary feature on the inside of the cell, but this could be a ring of actin, if you want to do that. Here this might be adapted somehow to the nucleus. Okay, so all I'm showing you here is the facility of this type of grid generation scheme to adapt to moving interfaces which could be contained in the intracellular region as well. Okay, now Charlie touched on this a little bit yesterday, but in generating grids, we also have to generate grids which are on the surface. So here I'm going to concentrate a little bit on curves. So the curve is going to be moving by a sort of force mean curvature. So I'm prescribing the normal velocity of the curve. Now that's enough to say how the curve is moving, but from a numerical discretization point of view, that can often lead to rather poor quality grids. So sometimes it's useful to get some control over the tangential velocity of grid nodes on an evolving curve or surface. So we're going to do this in a parametric form here. So we're assuming that normal velocity generally is some sort of curvature plus some normal driving term. But the velocity of the grid nodes are going to be in the normal direction with v, but in the tangential direction with some velocity b. So the question really is, what do we choose b to be? Okay, so in analogy with the grid generation scheme for the bulk, we essentially attempt to have this equidistribution property. So m here, again, so this is just a monitor function. This is essentially the rho I had previously. So this is large, where you want the grid spacing to be small. S here is the arc length. Sigma here is the parameterization. So we really would like that to be equidistributed around my grid. Okay, so if m was 1, I'd be satisfied with a grid which equidistributed arc length of the points. But if n was something which was not 1, I'd get adaptivity. Now again, I don't want to do this in an algebraic way. So what I'm going to do is I'm going to set up a PDE for the tangential velocity in the same way as the adaptivity I had in the interior grid. So I have an adaptive way of generating the grid on the surface that evolves with a given normal velocity. So here I want to show you, so here's again a test problem. This is an ellipse which is evolving due to mean curvature. And so basically what it's just going to do is it's going to collapse. But in the exterior region, I imagine I've got some traveling wave of some quantity that's passing over the domain. So simultaneously I'm getting adaptivity in the bulk, but I also have adaptivity of the mesh points on the moving interface. So there's a velocity in the tangential direction, which is sort of arbitrary, but it's attuned to this wave. And simultaneously, the points in the normal direction are moving with the correct mean curvature flow. So it's doing all of those things at the one time. Okay. So we can look at whether this is doing a good job. So here we can plot the area. Yep.
I have a question concerning the convective term in your concentration equation on the surface.
Yep.
So if I understand it correctly, you specify mean curvature flow with some forcing.
Yep.
That gives you only a physical normal velocity. And now you define a tangential velocity in order to just move your grid points and so on.
Yep.
But if you assume a cell which, well, let's say it doesn't change in its form, but just translates,
Yep.
it would be possible. So then there are always some points which does not move in normal direction, but only in tangential direction. So there should be a physical tangential direction.
Yeah, there could be. Yeah, absolutely. Yeah.
It's always there, right?
Yeah,
I mean,
yeah, absolutely.
So how do you get that? How do you incorporate this in your model?
So again, if there was a tangential physical velocity, that would be in the model as well. But
So how does it come in?
it would be in through the convective term that was there originally. The u, it wasn't assumed necessarily that originally was in the normal direction. So it could have a tangential component.
Where does this come from?
Where does the tangential component? Well, it would come from a model of if you were modeling the membrane, then you would have to model exactly where the displacement of those points were actually moving in the tangential direction. So with pseudopods are generated, the material velocity of the points tends to be in the normal direction. But in other areas of the cell membrane, they can be dragged along through tension effects in the tangential direction.
But even if parts move in normal direction, I mean, they're always like if this thing is translating the top and the bottom of your cell, it doesn't move in normal direction,
Yep.
it just moves in tangential direction. And just don't see how this is incorporated in your velocity.
Yeah, so there was a, yeah. So
The W is just the grid movement.
Yep.
The U is the physical thing, and that's the physical thing, and I don't.
so the physical velocity would normally come in through, you would have to have the physical velocity of what those material points were. If the velocity was in the tangential direction, you would have to have that from another part of the model. So you'd have to have a mechanical model that would tell you what those velocities were. And then they could be incorporated within the scheme.
There's a difference between the shape, material velocity, which is physical, and the velocity of the parameterization.
Yep.
They're three different things.
Yep.
To define the shape, you just need the normal velocity.
Yep.
May I comment? I think the answer to your question is just a pretty difficult one. It's modeling.
No,
I've
course,
thought
but-
about it a lot, and I have no answer yet. How to find the right.
Yeah.
But we agree that there's a tangential thing.
Yeah.
You need to
Right,
take
yeah.
account for the convection.
I completely agree. Yeah. The answer is hard.
Yeah, coming up with an appropriate U is difficult. But if you've got the U, then it gets fed into this quite easily. Okay, so so the question I had a little bit earlier was, so here's the simulated cell that's migrating at random here. So you can see the pseudopod's been formed, and this essentially is giving us a peak A, and this peak sort of splits. And the splitting gives us the two pseudopods which sort of move apart. And after a period of time, the action of the local inhibitor is that one of these tends to get quashed, and only one pseudopod seems to survive. And this leads to this sort of almost random migration, but you can clearly see that the new pseudopods are generated close to where the old pseudopods were. And even though there's no chemoattractant here, and if you do a number of simulations and calculate one of these so-called spider plots, so this is what happens in reality with Dicty cells. So interestingly, this almost looks random. Yep.
Sorry, to come back to the question of the oscillations then.
Yeah.
Is that because you're using Gira-Meinhardt? Is there just the oscillatory instability in there, or do you choose?
I think it's a combination of the fact that there's a combination of the domain shape feeding back on the solution of the reaction diffusion equations. So there's a two-way interaction between geometry and the reaction diffusion equations. So as I said, here's the so-called spider plot. So these are real cells, Dicty cells. These are the simulations. So okay. So you're seeing a little bit of determinacy, a little bit in here, but this is this left-right splitting, which on occasions gives you examples of a degree of persistency. And if you do a large enough number of simulations, you can calculate some mean square displacement data for the simulated cells and the experimental cells, and you do get this sort of persistent random walk behavior, where over a short period of time, these cells tend to want to move in the same direction, but over a longer period of time, they lose that information and they move more in a sort of diffusive state. So this is a simulation of the sort of micropipette experiment. So this is sourced here, and then we move it, and then we can see that the cell reorientates itself, but it doesn't do it by necessarily generating a new pseudopod exactly on the area on the cell membrane where it detects initially the highest degree of the chemoattractant. It does it by reorientating itself from let's say the head of the cell. Okay, so this is a simulation of the cell migrating where I'm actually solving my diffusion equation exterior to my migrating cell, and these are the grids that are being generated. So here, for efficiency, I also have a moving outer boundary, and we can see the cell again generating pseudopods, and we can see the interaction with the cell and the extracellular diffusive field. Okay, so this is chemotaxis up the way. Now, the sort of grids that you get, again, so this is chemotaxis in a linear gradient. They're nice quality. They're adapted nicely to the geometry of the particular cell. Now, Robert's Insol's got a very interesting problem recently. I was very concerned about looking at cells generating their own gradients of chemoattractants. So normally in chemotaxis, it's assumed that the gradients already pre-exists, and there's sometimes not a question of where it comes from. But this is an experimental setup here. These are melanoma cells which are migrating here, but they're migrating in the presence of something called LPA. And what's interesting is that this initially is a homogeneous background of LPA. There's no gradient. So what you're seeing here are the cells apparently displaying some degree of chemotaxis, moving from left to right in this sort of band here of cells. Okay, so it's thought that maybe what the cells are actually doing are locally degrading the LPA, thus creating their own gradient, and this is a mechanism for self-generated gradients. So, so it's relatively early stages for us trying to do simulations of this, but here we have a cell migrating, and you can see that the interaction of the generation of pseudopods with the external field is leading to a certain degree of persistency in the cell migratory pattern. So, so here we have a cell migrating away, and again you can sort of see here that what's happening is this is sort of leading to a sort of persistency in the cell migration. Again, where there's an interaction of the geometry, the receptor ligand binding mechanisms on the boundary, and the diffusion that's taking place exterior to the cell. And obviously, to verify this, one has to be pretty careful about the lengths and timescales of all of these different processes. You've got movement, you've got diffusion, you've got binding, and potentially in this particular situation you also might have enzymes on the membrane, and also they may be secreted in the extracellular region, degrading the ligand field. So there's a number of complicated processes that are all taking place in this time-dependent domain. Okay, so just some conclusions for the work. So hopefully what I've showed you is an adaptive of some application of some ideas from adaptive moving mesh methods to generate evolving bulk and curved meshes. Also talked a little about the coupling processes on and off the cell membrane using EL surface finite element method. And basically the role here is we're trying to develop good tools. I think someone mentioned it, I think in the previous talk, of good numerical tools for attacking these difficult real biological problems. It's already been mentioned that what's difficult in applying this sort of technology for intracellular problems is the specification of the material velocity field U. And also, given that, you may be in a position to include effects like focal adhesions to the substrate. And the other thing we would like to do is to extend the notion of these adaptive moving mesh PDEs onto evolving surfaces so that we have adaptivity of the meshes or the surface evolving in 3D. Okay, thank you very much.
So questions? John, you probably already answered that question, but the splitting of your maximum, is that the effect of local inhibitor, yeah?
I think it's a combination of the local inhibitor and the domain changing shape. If you take a circular cell and it's stationary, then you go into a steady state with this problem. The activator peak just sits there. Even though C comes up, it's not strong enough to kill the peak.
So you mean the fact that you start bending, making this...
The fact that you start moving, the fact that there's a very dynamic degree of curvature that's coming in through the membrane makes the whole thing much more dynamic. And that leads to one of the effects appears to be the splitting.
You had this very impressive this mesh outside of the cell, which you basically only need to compute the chemical distribution, right? So I agree that this is important when the cells consume or produce the chemical. In cases they don't, the cell might actually not perturb the chemical distribution much. So I guess you could just take a grid ignoring the cell, compute the chemical distribution there, and then just develop.
So that's what I said at the end, in the sense that one has to be a little bit careful about the time scales and length scales that are involved, especially because there's binding and unbinding that's going on. So that the effect that you were seeing in my model was purely the interaction of the geometry, movement, and receptor ligand binding. There could be it could be receptor internalization or enzyme degradation that would go on as well. So yeah, so if some effects are not as important, then you could certainly make some shortcuts.
Actually, he answered my question.
Okay.
We had some kind of very detailed assumptions, like for instance that the receptors are diffusible in the membrane. Is this actually, does this have any influence in your model, or are these kind of effects that you can ignore? Have you tried to?
It's not essential for the overall method that you have receptor diffusion there. But if all I'm saying is that's a complication that's dealt with in the numerical procedure. If you don't need it, you don't need it. It's not essential for stability. I didn't put it in there as a fudge factor or whatever.
Anyway, so these cells, they have like 50, 60,000 receptors. So
Yep.
you know, the numbers are so large
Yep.
that you would not really expect to be able to actually have to calculate changes in the distribution.
I have one question about the spider plot that
Yep.
you have shown.
Yep.
It looks like you have a strong axis effect on the trajectories, so they are going either 45 degrees or along the. Can you explain that? Is it just coincidental, or is it?
Yeah, that was just a subset of the total number of simulations that you saw there. If I plotted to actually get that mean square displacement plot, we had to do 20,000 simulations. Mm-hmm. If I plotted the 20,000 simulations, all you would see was just a cloud of these. So there isn't one particular preferred axis that somehow it's come into the system. It's just a subset of the ones.
Just one single trajectory looked like going like.
Yeah. So there's this business that it generates a leading pseudopod. It splits. So what tends to happen is it wants to then move in a direction. And the angle of that splitting has been measured in Victi cells, and actually our simulations do a very good job of reproducing that splitting.
Okay.
Then what tends to happen when it wants to split, it tends to want to split back in the opposite direction. There's a sort of 2-to-1 chance that it splits back to where it came from. And so that leads to this sort of persistence, essentially.
If no other question, thank you again.